I would like to invite Dr. Kathleen Murphy, who's the Associate Professor of Infectious Disease at Penn Medicine, who will talk to us about COVID-19 and give us an overview of the current state of affairs, as well as our future scenarios. Welcome, Kathleen.
Thanks, Dr. Agarwal. Hi, everyone, and it's a pleasure to be speaking with you this evening. It's always following Dr. Fauci is no easy task, but over the next 15 minutes, and I think that's perfect. Over the next 15 minutes, I will be giving a brief overview of the pandemic epidemiology, a clinical review of COVID-19 infection, as well as some additional updates on testing, treatment, and vaccination that hopefully will complement the information presented by others already, including Dr. Fauci. So to start, I would just like to again comment on sort of the global scale of the pandemic at this time. And it's really staggering to review these numbers. I know we've discussed them already, but we are now at the point of over 33 million confirmed cases of COVID worldwide. And specifically here in the United States, we have over 7 million confirmed cases and have just reached the sobering milestone of over 200,000 deaths since our first case diagnosed in January. This graphic is from a resource at Johns Hopkins University. The website's below. I encourage you to explore it. It's really an excellent compilation of data. This graph is also from the Johns Hopkins COVID Resource Center. And I'll just highlight here, this is a timeline of the pandemic as depicted by a 7-day moving average of daily confirmed cases, which is led right now, as you can see in the graphic, by India, the US, and Brazil. And the US, depicted in orange here, we've had fairly limited success in decreasing our case count, despite initial efforts in March of shutting down our communities and enacting social distancing. And as states began to reopen in May, you can see we had quite a second surge over the course of this summer. And I really think the trajectory of our curve from here on out is going to depend on a couple of different factors. We see schools and communities continuing to reopen. We anticipate challenges over the winter months with social distancing as more time is spent indoors. We are upcoming into the flu season, and how we can continue to encourage social distancing, masking, testing, and contact tracing is all dependent, as well as a safe and effective vaccine. So there's a lot of things in play to where we go from here. So now I just want to talk a little bit about some of the clinical aspects of COVID-19 that I think are important to review. As Dr. Fauci touched on, one of the challenges of the SARS-CoV-2 virus is that a patient's peak infectivity may actually be within the 24 hours prior to symptom onset or right at symptom onset, which is really important in terms of mitigating transmission and creates quite a challenge. And as was mentioned, up to 30–40% of people are asymptomatic or have very minimal symptoms, yet can still be significant vectors of transmission. I agree with Dr. Fauci that there's likely a spectrum of transmission. And as you probably have read in the scientific literature and in the media, there is quite a debate right now over transmission dynamics between droplet and airborne spread. But I agree that the most prominent form of spread appears to be via droplet transmission, although we do worry about airborne transmission, particularly in very close confined spaces with poor ventilation. And I think that's a particular concern in some situations where we've seen some outbreaks stem from. But one thing that does reassure me about the mode of transmission predominantly being droplet is the efficacy that we've seen with universal masking. And that's particularly been prominent in my role as a hospital epidemiologist here at Penn, where when we enacted universal masking in March, we saw a significant decline to essentially no staff-to-staff and staff-to-patient transmission with universal masking. And we've been very, very encouraged by that data and our personal experience here. As was mentioned, we have a better understanding now about the spectrum of clinical presentations of COVID-19, which I'd put some CDC data from their surveillance network here on the right, similar to mentioned by Dr. Fauci. We know about 20% of patients will require hospitalization and about 5% ICU level care. And our overall mortality rate right now in the US is about 3% since the start of the pandemic. But we've made significant strides within our critically ill population relative to how we ventilate patients, anticoagulation, and some treatment strategies, including steroids that we'll touch on in a little bit. We know about risk factors for severe illness, which have already been highlighted, including an amazing review by Dr. Agarwal of what's known in the oncology population, although we need more data, including in organ transplant and other immune compromised groups. And we really do need more studies about long-term sequelae. Here at Penn, we have a post-COVID clinic run by our rehab clinic and our pulmonary doctors, and we're seeing a lot of chronic respiratory complaints, fatigue, and cognitive impairment in patients that we need to understand and look into further. And I'll also comment that we have seen an incredible disproportionate impact on our Black, Hispanic, and Native American communities, as was mentioned by Dr. Fauci. And it's really highlighted the urgent and important efforts need to address longstanding systemic health and social inequities. So I wanted to go a little bit more in detail into testing, as has been asked in our question and answer session already. We have three general types of tests for SARS-CoV-2 at this time. And there's multiple platforms that have been designated and granted approval by the FDA for emergency use authorization. Molecular testing has by far and large been the most common test performed. We've performed over 90 million of these tests in the US to date. It's most commonly used in the healthcare setting. And this test uses reverse transcription PCR testing to essentially detect SARS-CoV-2 RNA from respiratory tract specimens. They are very analytically sensitive and very useful in high-risk settings such as hospitals or nursing facilities, dialysis units, where it's really important to have a very sensitive test so you can contact trace and isolate people appropriately. These tests have a very low positive rates. We have seen challenges with false negatives, even though this is a highly sensitive test, if patients are tested too early in their course, related to specimen quality. And we do think there's some discrepancy between upper respiratory tract and lower respiratory tract shedding. And it's not always feasible or safe to get a bronchoscopy, for example, or an induced sputum sample for diagnostics. One of the biggest challenges with molecular testing that I get asked about a lot is just a reminder that it doesn't differentiate between live or infectious virus or continued shedding of sort of dead viral genetic fragments. So it's really not a test of infectivity. And patients can continue to shed virus for weeks to even many months after their initial diagnosis. And we still have a very limited capacity of rapid molecular assays, which impacts us on a daily basis in the hospital. Antigen-based tests, which detect viral protein, are also increasingly available. And these are attractive because they're rapid tests, they have results in minutes, they don't need extensive lab equipment to perform them, and they are less sensitive than molecular tests. But I do think they have a role because of their low cost and ease of administration, particularly in low-risk settings where you're doing surveillance, for example, in school or university settings, or for other population-based screening efforts. Antibody testing is now available, which most often detects IgM and IgG to the SARS-CoV-2 surface spike protein, which has been described by Dr. Fauci. And people develop antibodies usually between 1 and 3 weeks after symptom onset. The sensitivity and specificity can vary based on assay. And we need more data about the duration and actually role of antibody-mediated immunity. Some studies have shown that antibody levels start to wane even within a few months after infection, and we need more information on that. I'll just highlight here, and someone had submitted a question actually about saliva-based testing, but I'll highlight this study from Yale, where researchers actually validated saliva-based molecular testing and found actually higher amounts of SARS-CoV-2 RNA in saliva specimens and less variation over time than compared to the traditional nasopharyngeal swab. And so this technology was actually used by the NBA this year for surveillance testing, and Yale's protocol now has been granted an FDA EUA. Advantages of saliva-based testing, which are very attractive to me, is that people can self-collect samples. We don't need to rely on swabs or other transport media, which has had shortages already in the pandemic. And for all these reasons, we're also currently validating a saliva-based test here at Penn. In line with discussing testing strategies, I think it's important, because we've also struggled with this during the pandemic, just to comment on the challenges of using testing to determine when a patient is no longer infectious and recovered from COVID-19 and can come out of isolation. And in general, at the beginning of the pandemic, we were using mostly a test-based strategy where we needed to see 2 negative tests to clear people from isolation, to now more of a time-based strategy where we wait for a duration of time after people develop symptoms to clear them in terms of from isolation and either in the hospital or at home. And as I mentioned, the challenge is that molecular PCR tests can, again, remain positive for weeks to months after initial diagnosis and can just reflect dead viral genetic material that's still in the respiratory epithelial cells. And if we wait for these test-based clearance, this has resulted additionally in delays in care, and it's really challenging logistically to coordinate multiple repeat tests. So the CDC guidance has now shifted to, for most patients, except for severely immunocompromised patients, to move towards a time-based strategy. And the data we currently have looking at actual culturable virus from respiratory samples indicates that there's no cultural virus between 10 and 20 days, depending on illness severity after symptom onset. And I think relative to the oncology population at Penn, we've really worked closely between infectious disease, infection control, and our oncology providers on how to navigate individual decisions for cancer patients, for example, when to return to treatment or start other immunosuppressive agents. It's very similar in the solid organ transplant world, and we need more data on duration of infectivity in immune-compromised patients so that we can more accurately rely on time or really use a test-based strategy in this setting. I'll comment now just about briefly on some treatment options. Some of these studies were already mentioned by Dr. Fauci, so I'll leave the information here and just touch briefly. But one of our most promising treatments is dexamethasone, and we really think hopefully this mortality is related to inflammation-mediated lung injury that occurs a little bit later in illness course. But this is data on the recovery trial that Dr. Fauci mentioned. And as was discussed, it showed a significant 28-day all-cause mortality benefit for patients who were on oxygen, particularly in ventilated patients, with the caveat that patients with a longer duration of symptoms had the greatest mortality benefit. So we do think that there can be harm to giving steroids too early. Remdesivir, which Dr. Fauci also discussed, is a viral RNA polymerase inhibitor, which has good in vitro activity against SARS-CoV-2, so has been promising. Now has FDA EUA approval as well for all hospitalized patients. And in May, the ACT-1 study, which was the adaptive COVID treatment trial, was published in the New England Journal that showed a faster median recovery time by about 4 days for patients receiving remdesivir, although no significant mortality benefit at 14 days. And we also have a better understanding now about duration of treatment therapy. For non-ventilated patients, there seems to be not a benefit to using longer therapy between 5 and 10 days. And so remdesivir, particularly in the past, has been a limited resource. And so that's helped us navigate our dosing strategies. I'll speak a little bit about convalescent plasma, which has received a lot of media attention recently and is now also has FDA EUA approval. Unfortunately, we need more data. The current randomized control trials that we have not shown a mortality benefit, 2 of which I've listed here by Li et al. Published in JAMA, which was an RCT in Wuhan, China. And then the PLACID trial just recently preprint revealed their data. This was done in India, again, with no mortality benefit, but still in preprint and needs to be peer reviewed. The Mayo Clinic has published information on their experience with convalescent plasma. They were originally providing plasma through an expanded access protocol, so have a cohort of about 30,000 patients who received plasma. And just looking at their cohort of people who received plasma, they did see a mortality benefit to people who received it earlier in disease course and who received higher titer. But again, this was all compared within their group of patients who had received plasma. So we need more randomized control trial data. So at Penn, we've worked with our antimicrobial stewardship team to really come up with a guideline for our patients, which follows a lot of the NIH guidelines as well. We use remdesivir for hospitalized patients with moderate or severe illness, dexamethasone based on, again, oxygen requirement. We have a trial that we can enroll convalescent plasma, and we're looking into other immunomodulatory agents in the trial setting. We're enrolling patients in a trial looking at baricitinib here, but we need more data on immune-compromised patients for sure. And then finally, I'll just speak to vaccines. So this is a website by the New York Times that has a really great up-to-date review of vaccines and where they are in development that I'd encourage you to look at further. I think that the question of when will have the vaccine is difficult to answer, as Dr. Fauci alluded to. And we really need a commitment to a transparent evaluation of safety and efficacy. You know, as healthcare providers, we are going to need to be comfortable being advocates for vaccination. And a lot of this depends on how transparent the data is, and really to generate public trust in the vaccine. Here's a list of what I think are 4 of the most promising vaccine candidates in phase 3 trials. 2 of them are mRNA vaccines, Moderna and Pfizer, and the other 2 are adenoviral vector vaccines, all currently in phase 3 trials at this time. And the interesting thing about the vaccines is that in sort of an unusual move, these 4 manufacturers have actually published their phase 3 trial protocols. So that's given us a little bit of insight into the data we can expect. So all are enrolling target enrolling tens of thousands of patients. 3 of the vaccines, all but Johnson & Johnson, require 2 doses, which is sort of important to think about broad scale operationalizing of this. And then 2 of the vaccines, the mRNA vaccines, require storage in ultra low temperature, which has also led to some consideration of challenges of how we would coordinate and transport these for mass scale vaccination. But I'll just leave with a comment that, interestingly, the primary endpoint of these vaccine studies is important to understand because they're all looking at prevention of symptomatic COVID-19 infection, which can be mild disease. Only the Johnson & Johnson primary endpoint includes severe or critical illness. So we just need to take this into account because ideally the vaccine would do more than prevent mild disease. Ideally, we would have time and data to really show prevention of moderate and severe illness. And I think when this data is available and how the FDA will navigate their criteria for expedited approval is still to be determined. And I'm also eagerly awaiting this data. So I'll end there. Thanks for your time.
Thank you so much, Dr. Murphy. That was an amazing overview of the current epidemic or pandemic, I should say, the current available testing and upcoming vaccine strategies. Thank you so much again.